Hello, everyone. So we are moving forward with our theory materials. In this recording, we will be discussing chemical reactions as well as enzymes. And I would like first to go over a couple of slides, which some of us covered very, very quickly in class last time, while others didn't have enough time to go over those. So we'll cover them right now for the benefit of everyone. So the question we're asking ourselves after we've seen the different macromolecules and organic molecules in general, so we want to know how cells are capable of making or synthesizing so many different organic molecules, right, that have many different structures and many different functions. Well, the answer to this is chemical reactions, right? Remember that cells are biochemical factories, and so using special machines that we call enzymes, they're able to catalyze a large variety of chemical reactions. For example, if you take here methionine as a starting building block as an amino acid, which the cell would already have, it's able, through the use of some enzymes, which we can see here. So enzymes typically end in A's, right? So you can identify them that way. Through the use of some enzymes, it's able to convert one molecule into another, and then into another, and then another, eventually to obtain a product that's going to be useful for that cell. So in this case here, ethylene, which is a plant hormone, is produced starting from the amino acid methionine, as well as using some ATP and some other substances as well. And then ethylene has various effects for plants, such as causing fruits to ripen, to mature, and the shedding of leaves, and so on. If we look at another example, here in animals, steroidogenesis, so steroid, and genesis means creation. Okay, so creation of steroids. Well, here what we can see is that starting from our initial building block, cholesterol, okay, which is either obtained from food or produced by animals, that is, so animal food, or produced by the animal itself, such as humans. We produce our own cholesterol. So we start with cholesterol, and then through a series of chemical reactions, we're able to produce various hormones, such as testosterone, estrogen, cortisol, aldosterone. So you must have heard of some of those, progesterone. And so we start with cholesterol. Then we have one enzyme here that's going to convert cholesterol into another molecule. In this case, it's cutting a piece here. And then next, you're going to have another enzyme here converting our our molecule here that has been already converted from cholesterol, converted further. And if you look carefully, you would see that what we're doing mainly is just adding or removing functional groups, right? And those functional groups then are going to be important for that molecule to interact with other molecules. So we can go through the different steps. So in a cell, if you have all those enzymes, then you would be able to make all the proteins that are listed here. Okay, so we have stepwise modifications, always through the help of an enzyme, which speeds up or make possible the chemical reaction to occur fast enough. There's often several possible routes to take to reach the certain desired product. And also something to think about is that not all cells will necessarily produce all the enzymes, right? Those enzymes in the red and green boxes, um, some of them will be produced in some cells and others might be produced in other cells, right? So for example, a cell that produces estrogen or estradiol will need this enzyme here, aromatase, right? convert at some point. It has some testosterone and it converts it to estrogen. While a cell that makes cortisol would not need that enzyme, but instead would need this enzyme here. Okay, so different cells are going to use different enzymes so that they can produce the desired products, right? So estrogen would be made in the ovaries, for example, and cortisol would be made in the adrenal glands and in humans or in animals. Okay. So this is a biochemical pathway or metabolic pathway, but what we can see is that we're able to make many different molecules, slightly different each from each other, that will have different functions then. Here's another example. Starting from tyrosine, which is an amino acid, we're able to make various molecules, such as dopamine, which you might have heard. It's a neurotransmitter. So this is produced in the nervous system, in the brain, spinal cord. So if we're talking about a neuron, well, the neuron would need to have this enzyme and this enzyme to be able to make dopamine. But then if we're talking about if we want to make epinephrine, that's adrenaline, same thing. For example, we want to produce that in our adrenal gland. Well, we would need to have all 4 enzymes to be able to start from tyrosine and to make epinephrine. Okay? So if we were to map the whole set of chemical reactions that can occur inside a cell, you see that it becomes quite overwhelming. There's many different, so each connection represents the transformation of one substance to the next, and then the lines represent the chemical reaction. So you'd see that there's a lot of different chemical reactions that can occur, and they are all connected, right? So what our cell would like to do, any cell, is to make the specific molecules it's interested in. So it's going to need to, turn some switches on and off to kind of direct the flow of chemical reactions that is taking place. Okay. All right, and now on to the new material regarding chemical reactions and enzymes. First, we'd like to think about physics, right? We're scientists, so we link the different scientific disciplines. So if we think of the laws of thermodynamics, we know that energy is conserved. Energy cannot be created or destroyed. Energy can be transferred, though, or transformed, can be converted. For example, we can convert potential energy or chemical energy that is found in the bonds of the molecules that, well, from that animal that the cheetah is eating. So that's chemical energy. And that chemical energy then, through some metabolic processes inside the animal, will be converted to kinetic energy, right, when the cheetah is running. And kinetic energy, that's movement. And also keep in mind that when this occurs, when energy is transferred, the entropy can increase. So if we're losing or releasing some of the energy as heat, then we're increasing the entropy in the universe, actually. So that's the second law of thermodynamics, entropy. That is, energy transfer or transformation increases the disorder or entropy of the universe. Okay, so something you might have seen in your physics classes. Here are some other examples of energy transformations. Again, chemical energy to kinetic energy. So animals would obtain their energy from food, right? Chemical energy. Plants, they obtain their energy from light, but they're going to convert that light energy using some matter, using CO2 and water. They're going to convert it to chemical energy, right? Where we have glucose molecules that can serve various purposes. They can be used for for storing the energy, right? The potential energy. They can also be used to provide structure to the plants. And here's just another example. If we think of gravity as well, right? So you have more potential energy when you're higher up. And so, yeah, and when you jump, then you release some of that energy that's converted to kinetic energy. And if you want to go back up again, then you'll need to expend additional energy. Okay, so you're going to convert this kinetic energy to potential energy. Now that's not in the bonds of the molecules, but that is in the position of the individual. So something to keep in mind, especially when thinking about biological entities, is the difference between open and closed systems. So here we have actually a very closed system or isolated system where we are not able to transfer any energy in the form of heat, and we're not exchanging any materials either, no mass and no energy that is exchanged with the outside. That's an isolated system. Then we have a closed system where energy can be transferred in the form of heat, but mass cannot. Okay, and finally we have an open system where both energy and mass can be exchanged with the outside. So if we look at some daily life examples, if we think of the thermos here, it resembles which type of system? Yes, isolated system, because we're not transferring heat, or at least very little, so mostly isolated. Then we have our bowl of soup here. That would represent a closed system, right? Because we can exchange heat, and eventually the temperature will equalize between the inside and outside. But we're not exchanging materials. And lastly, our open system, our bowl of soup is the system. It exchanges energy with the outside, and it also can exchange some materials. In this case, we're adding some stuff in our soup. Now, when you think about biological entities, cells, organisms, right? For example, the cheetah that we looked at before, are these open or closed systems? Yeah, they're open systems, right? And you might think, well, biological structures are very ordered, and we're maintaining that order, and we need constant energy input to maintain that order, but we have low entropy in our biological entities. So how does that not violate the law of thermodynamics? Well, although entropy in our biological system is low, we're increasing the entropy in the universe, so the outside becomes more disordered, more disorganized as our biological entities become more organized. Okay, kind of interesting to think about. Okay, so going back to the cell as a biochemical factory, a theme that we've seen before, keeping that in mind, we know that in a cell we have inputs, right, in the form of building materials and energy, and the energy can be from nutrients, that is from matter, right? And in this case, sorry, nutrients, let me redraw this. And in this case, we're talking about potential or chemical energy, right? That's found in the bonds of those nutrients. Or if we're talking about animals, animals will obtain all their foods through, all their energy through nutrients, or it can be through the sun, right? So for example, plants. So we've got inputs, we've got some processing of those inputs, and we make some outputs, products that will be useful to the cell or to the organism, as well as some wastes. And we also release some heat, so we're increasing the entropy, right, in the universe that way. Now, the question is, how do we process those inputs to make those outputs. So we're going from original building materials to other molecules, other structures. And so how do we do that? Well, we know that we do it through chemical reactions, right? So that's what we're going to discuss next. So when talking about chemical reactions, we can classify those into two categories that we call endergonic or exergonic. So the endergonic reactions, in those reactions, the products that we make have more energy than the initial substrates. Okay, so if we're, for example, building a larger macromolecule out of smaller subunits, right? We're adding covalent bonds to join those subunits together, and we're increasing the amount of energy, sorry, that is in the products compared to the substrates that we used. So that's called the delta G, and that we will have a positive delta G. That's also called Gibbs free energy. And so we say energy is absorbed, and we can understand that the reaction is not spontaneous. It's not going to occur on its own. Now, we also have exergonic reactions, and there is the opposite. That is, the energy found in the products compared to the energy from the reactants is lower. Okay, so again, maybe you had a macromolecule that you broke into smaller subunits, and therefore we have less energy in the products. Delta G, Gibbs free energy, is then lower than 0. All right. We say that energy is released. And in this case, we say also the reaction is spontaneous. It will occur on its own. However, in order for any chemical reaction to occur, you need first to expend some energy, because you need to be able to convert one state of molecular structure into another one. So we need to kind of destabilize those molecules enough so that the reactions take place. So that's called activation energy. In both endergonic and exergonic reactions, we need activation energy. Yeah, rate of chemical reactions is decreased by increased temperature and increased substrate. We'll come back to that later on. So, okay. Now, the problem with this activation energy, if I go back, actually, you see that it's quite a steep curve. To be able to reach that point, it's going to require quite a bit of energy. And yeah, if our temperature was very high, then we'd have a lot of molecules that are moving very fast and have a lot of that, enough, sufficient activation energy for the reactions to take place. But in biological systems, we have relatively low temperature, and substrate concentrations are also relatively low. So we cannot really wait for those chemical reactions to occur. They will be just too slow for life to be sustainable. So how do we allow chemical reactions to occur then in biological systems? We need to lower the activation energy. It has to be easier for the reactions to occur. And how are we going to lower the activation energy? Any guess? Yeah, through enzymes, exactly. Okay, or catalysts, right? Enzymes are catalysts. So by adding an enzyme, what we do is we lower the activation energy so that it's easier for the reaction to occur. Now, keep in mind that this does not change in any way the amount of energy that the reactants and products had. So if we had an endergonic reaction, we still have increased the amount of energy that is found in the products compared to the reactants by the same amount, okay? But it was just easier to get to that new state of molecular organization. Same thing for exergonic reactions. As you can see, regardless if you have an enzyme or not, we're going to have still the same energy, in this case, that is released, free Gibbs energy released. But you see, it was easier still to perform that reaction with the enzyme. The curve was lower. Okay. So here's just another example. Let's say you want to break lactose into glucose and galactose. If you just wait there and let your lactose break down, it's going to take a long time, and that will not occur on its own, for being meaningful in biological systems, right? It would be too slow, because we need to expend enough energy to break that bond, and we cannot just increase the temperature so high, right? So with an enzyme, we're going to decrease the activation energy, allowing the reaction to take place fast enough. Okay, so again, enzymes lower the activation energy. There's no change in the free energy that is absorbed or released. You can think of enzymes as tools, right, that just make it easier to perform a certain task. And here you see also a little ball that is jumping, and it has to jump to the other side. So with an enzyme, we just lower the bar, right? We make it easier for the reaction to take place. So let me show you a little video that describes exactly this thing here.
Envision a molecule, here symbolized by the ball, that in principle can react in 4 different ways. To undergo any of these reactions, the molecule must overcome an activation energy barrier that is of a characteristic height for each of the possible reactions. This can be achieved, for example, by putting more energy into the system, such as when heat is added. In the example shown here, the molecule will enter many different reaction paths by overcoming similar activation energy barriers. An enzyme, in contrast, reduces the activation energy barrier of only one specific reaction path. An enzyme, therefore, allows reactions to proceed at normal temperatures and directs them into one desired pathway.
Okay, so hopefully that makes sense. Again, we cannot just increase the temperature in living organisms. Instead, we need a different strategy. So enzymes are the way to go. So how do enzymes do it? How do they lower the activation energy? Well, keep in mind that if we didn't have enzymes and we let molecules just move around, right, with their kinetic energy, they're just moving in all directions randomly. And once in a while, 2 molecules are going to bump into each other. However, in order for a chemical reaction, they must hit each other with enough force and also in the proper orientation, such that the functional groups involved in what would be the chemical reaction. So let's say you have a functional group here and functional group here. They must align so that the reaction can take place. Well, if we just let the molecules move randomly, most of the collisions between those molecules will not be productive. That is, they will not result in a chemical reaction. So we're going to try to use enzymes to facilitate this process to have productive collisions, and that is chemical reactions resulting. Okay, so what the enzymes do? Well, one thing they do is they will bring 2 substances in closer proximity. So first step, right? You want to bring them closer together. And if the enzyme is able to hold on to those 2 molecules long enough and they're close together, a reaction might occur. Even better, not only would you want to keep those 2 molecules together, but you would want to orient them such that it makes it even easier for that chemical reaction to take place, because the functional groups will be very close to each other. Okay, so this is basically how the enzyme would do it. Here we have another example. And this is more detailed. It's nice to see here, this is our enzyme, right? And our enzyme, you know that it's a chain of amino acids. So you see here in blue, yellow, and green, these are the different functional groups of the amino acids. Those functional groups are important to form the 3D structure of the enzyme, but also some of those functional groups are present, allowing to interact with a substrate. Okay, so those ones in yellow here. And we have another image just showing the same thing, but kind of nice to distinguish between the polar groups, non-polar groups, and charged groups. So these are present in what we call the active site of the enzyme, where the substrate will be able to bind. Okay, so you can imagine that polar will bind with polar, or will interact with polar. Charge will interact with opposite charge, and non-polar would interact with non-polar, or would arrange itself closer to non-polar parts of the substrate as well. So here, when we have the molecules added, you see that the enzyme kind of allowed to anchor those molecules in place, and now they're close together. So there's a much greater chance that they're going to react with each other. But we can go a little further if we're able to just twist a little bit that enzyme molecule and get those two parts even closer with their functional groups, then the reaction will very likely occur, right? So if we change the shape of the enzyme just a bit, those two parts here get closer together and the reaction takes place. By the way, pay attention to the functional groups on the enzyme and substrate and see how they match. So you see here the polar groups are matching, so probably forming hydrogen bonds in between them. We have the charge parts of the molecules as well, positive and negative, attracting each other. We have the hydrophobic sections of the enzyme and the substrate, which would tend to cluster close to each other as well. So this way, really, the enzyme is going to form a bond. It's not a permanent bond, but still, it will allow, again, anchoring of those substrate molecules and positioning so that we facilitate the chemical reaction. Now, when we think about the enzyme-substrate interactions, we can think of some models that we often discuss. One of them is the simple lock and key model, where the substrate is the key that binds to the lock on the enzyme's active site, and it's a perfect match, right? So you see perfect lock and key interaction. So this model is useful to think about how enzymes and substrates interact. However, a more realistic model is the induced fit model. That is, as the substrate gets closer and closer to that pocket here, which is the active site, there's going to be a slight change in conformation in the active site to better fit the substrate, and then the substrate will fit in that location, right? Now, interestingly, after the substrate moves away, maybe after the chemical reaction has occurred, or maybe it could also just detach, the original shape of the enzyme will be restored so that the enzyme can once again perform a chemical reaction. So yeah, when we think about enzymes, the enzymes are conserved, right? So although a chemical reaction occurs where the enzyme changes shape slightly to facilitate the chemical reaction to occur, eventually it goes back to its original shape. And therefore, it's available to catalyze another reaction. So that's why we like to talk about the catalytic cycle of an enzyme, because that's going to continue again and again and again as long as the substrates are present. And yeah. Okay, just a couple of terms that you will hear about at some point. Some enzymes work on their own, but some other enzymes will require some additional help. They require cofactors or coenzymes, and sometimes there's some confusion about those terms. But basically, in general, a cofactor is a molecule that is going to bind to the enzyme to make it easier for the chemical reaction to occur. So you can think of it as an activator. Often the cofactors would be inorganic, but in some cases, the cofactors are also organic. And when we're talking about organic cofactors, we call those coenzymes. And these would bind, typically they would bind to the active site to provide a proper shape for the substrates to bind. So basically they help recruit the substrates and have them positioned properly. Okay, so cofactor and coenzyme are often needed for the enzyme to be functional. So let's summarize some important properties of enzymes, most of which we've seen in previous slides. So it's good to know that most enzymes are proteins, as we have seen before, but there are a few exceptions, and these involve RNA. So some RNA molecules have catalytic properties, so they act as enzymes. We call those ribozymes. Interesting to think about that. Enzymes may require cofactors, coenzymes, as we've mentioned before. Enzymes are highly specific. They're going to act on a single substrate or a group of related substrates that would all bind at the same active site. So they would have similar shapes at that location where they bind. Enzymes are going to allow for much higher reaction rates than would occur without them. So they're going to make possible life, basically, right? That they're making possible chemical reactions at a rate that is viable for cells and organisms. So if we look at a couple of examples, or at least this one here with peroxide. So if you were to leave your peroxide bottle for months or years, eventually you would have some of those peroxide molecules that would break down to water and oxygen, but that's going to take a very long time. If we were to add iron ions as catalysts, we could increase the rate of reaction by 30,000. So that's already good. But now with the enzyme catalase, which is shown here, we're going to increase the speed of chemical reactions by 100 million, much, much faster. And by the way, catalase also possess some iron atoms embedded in its in the enzyme that will also contribute to making the reaction faster. Enzymes may be used and reused to carry out the same chemical reaction over and over again, right? That's the catalytic cycle of the enzyme. It's often shown with this equation here, enzyme plus substrates. There's here a temporary conformation where the enzyme and the substrate are attached together. Then the reaction takes place. We have a product detaches, and the enzyme can be used again with another substrate molecule, right? Enzymes will allow chemical reactions at milder conditions, which is necessary for life. So for example, temperature and lower temperatures. Finally, enzymes have capacity for regulation. Okay, so we're going to look at that next.